All right, so today we're just going to refresh your memory about transcription, translation, and maybe tell you some things that you did not know or forgot that I told you in previous years. So anyway, transcription translation, that's a process that we use to take DNA and make mRNA, and then mRNA to make proteins. So remember that the purpose of DNA is to direct protein synthesis, and then proteins do the work of the cell and pretty much make everything else that we need. So a couple things: transcription, right, the synthesis of RNA under the direction of DNA. So that's what I mean. So the instructions for protein synthesis, that's the DNA. Messenger RNA will then carry the genetic message. I lost my pen here. Will carry the genetic message to the ribosome for the synthesis. So, as we talk about biotechnology, two things that are really important is that in prokaryotes or bacteria, mRNA makes, or transcription makes mRNA. So if you look at our little crude picture of a bacteria here, as the DNA is being transcribed, right, it's making proteins from ribosomes right at the spot of transcription. So as it's being transcribed, it's already translating it to make a protein. In eukaryotes, right, it makes pre-mRNA. And the reason it makes pre-mRNA, because we have the presence of introns, remember. Right, so we talked about last chapter, if we were to place DNA into bacteria, what kind of DNA would we put in there? Right, so if I had a human gene put in here in this bacteria that I would want to have bacteria make a protein for me, then the type of DNA I'd want to put in there would be cDNA, because cDNA lacks those introns. Okay, so the reading frame indicates a proper start and stop location. Translation is synthesis of protein under the direction of mRNA. So we're going to talk about both of those things today. All right, so let's get into it. So the genetic code. Remember, the reason why we're able to make proteins in bacteria that we can use in humans is the genetic code is universal for all organisms. Right, so we have this little box here, we have the wheel, one of the two. But remember, there are 64 codons and 20 amino acids. It's called a redundant code because when you look at these, if you look at down here at a valine, right, there are a whole bunch of different combinations that'll make valine, right? And that might be a leftover of evolution and slight mutations and the ability of organisms then to use that code to make similar proteins. So it's called a redundant code. There are 20 amino acids, right? Things that you probably remember, AUG is the universal start codon, right? So, and then we have 3 stop codons, UAA, UAG, and UGA, right? So those are my stops. All right, so that becomes important later on. But remember, bacteria can make human proteins because that genetic code is the same. All right, so transcription. First start. We talked about this in a chapter earlier. First thing, right? So we have this promoter region. Right, RNA polymerase will probably part DNA, right? And that DNA, remember, those are in eukaryotes. I don't want to write that. In eukaryotes, that will be in euchromatin, or that chromatin that's loosely packaged around histones, right? So RNA polymerase will probably impart a section that needs to be read, right? So in DNA synthesis, that would be helicase, but RNA polymerase is able to do that by itself, right? It will look for the promoter region, right? It'll read the DNA in 3 to 5, and we've talked about that so many times, and that's called the sense or the template, right? And it will start at AUG. So the first codon it's going to read is AUG. codons are in the 5 to 3 direction. So on DNA in the template stand, that would be TAC, right? So it'll bind to the promoter at the TATA box, right? So if you look at our promoter region here, right? So this is our promoter region, and this TATA box, right, aligns at RNA polymerase, so it can start to move along and look for AUG. Once it finds methionine, it starts to transcribe, and it goes in that direction, and it'll keep making transcript until it reaches a stop codon. Right. So this should be a review for you. Right. Transcription occurs, like I said, until it reaches stop codon. Right. Remember, the codons are UAA, UAG, UGA. You might want to jot those down. And when it reads those, right, when it gets to a stop, RNA polymerase falls off the DNA. Right. So protein synthesis will begin at the promoter, and it'll start, and it'll hit until it hits this terminator. Right. And that terminator will be the UAA, the UAG, or the UGA. Right. So my template strand was 3' to 5'. My pre-mRNA will be in the 5 to 3. Right. So after that pre-mRNA is made, remember it contains exons and introns. Right. So exons are coding regions. Introns are NTR regions. Right? So the pre-mRNA has to be modified. So I have to get rid of those NTR regions. Right? So this region I have to be able to get rid of. So splicing occurs. Generally speaking, there's a group of small nucleotide RMPs. It's a type of RNA. The group of these will form what's called a spliceosome. And that spliceosome will remove it. And the way it does it, I've had my DNA here. It'll form these loops, and it will join this section right here, and it will remove the intron. So it forms these loops, it removes the introns, leaving only exons. Right, so that's what that happens. And then at the end, you get a GCAP here. And that GCAP consists of GTP. Right, and remember we talked about at one point that GTP could be used kind of like ATP to power a reaction. Right, we use that for surface proteins a lot. And then a poly A tail. So that's just a long sequence of adenines, and this helps so both of them help with resisting degradation, right? They protect the ends, but they also have different functions. So the GTP powers initiation, right? And the poly A tail can help hold the reading frame as well, right? So once all those introns are gone, I've got my GCAP, my poly A tail, then I have mRNA. Okay, so all that has to be done with eukaryotes. So one of the benefits of having prokaryotes make proteins for us is that we don't have to worry about a GCAP, we don't have to worry about a poly A tail, we don't have to worry about removing NTR regions. As it's cranking out the transcript, it's translating, it making proteins. So there's a benefit of it. The complexity of the proteins is somewhat limited. So for some sorts of small proteins like insulin, it's the perfect way to go. But some of the larger proteins that need specialized structures on them, polysaccharides, whatnot, it doesn't work as well. So that's why we have things like CHOs. Right. So tRNA, right? So mRNA is a transcript of bring. tRNA contains the anticodon, right? And the anticodon is the complement. Oh, I forgot to mention, I probably should mention, right, that you need to know what side these are on. So the G cap is on the 5' side, and the poly A tail is on the 3' side. Right, so the poly A tail is added to the 3' side of the transcript. Lost my pen again. Right, the 5' end. Okay. All right, so the tRNA contains the anticodon. The anticodon is the complement to the mRNA. Right, so the tRNA are always present in the cytosol, and they have complementary amino acids attached to them. Right, so if you remember that there are 64 codons, right? 20 amino acids, so there are 61 codons that code for amino acids and 3 that are stops. But we have about 24 tRNA, right? Because many tRNA can handle the complementary amino acids that have different transcripts. And we'll talk about that. So initiation of translation. So translation occurs when that small ribosomal unit binds first to the mRNA. So if that small ribosomal unit will find that mRNA within the nucleus of a eukaryote, and it will start to exit through the nuclear pores as the large unit attaches as well. So first thing that happens with initiation is a small ribosomal unit will bind to an mRNA, right, and search for methionine, right? So if we have the small unit, I'll draw it here. The small unit has 3 sites on it, E, P, and A. And if we have a mRNA right, so if we have our mRNA, and this is my 5' site, remember, right? So I have my G cap, my thionine, right, is my first. So my small unit, right, will search this, and it'll lock methionine, right, or AUG at the P site, right? So once it does that, then the large unit will attach on top, facilitated by GTP. My GTP will exit and become GDP plus PI, right? So I use that energized phosphate to help power this binding of these 2 units. And at that point, translation can begin, right? So, like I said over here, the large unit attaches after AUG is found, completing the initiation complex, and translation begins. Right, there are other things called transcription factors that help bind them together. But essentially that's how it happens. And this whole unit will exit through one of the nuclear pores. And if the mRNA is made to make proteins for the cytosol, it'll remain in the cytosol. And if it has what are called transcription factors that identify it to be a membrane protein, then it will go to the ER. And we'll talk about that in a second. So mRNA then stay consistent. We'll move in the 5 to 3 direction. All right, so now I've got my big unit here, and I've got my A, my P, and my E site. Right, AUG, methionine, is attached to my P site. And then this transcript will just start moving in that direction to the ribosome. Right? So there's the 3 sites there. My next amino acid will come enter my A site. Right? So the A site is for attachment of the next tRNA. And then my E site, I lost my pen again. My E site is for the exit. Right? So this will come in and it will attach right to the growing chain there. This bond between here, remember it's going to be called a peptide bond. Right? And then my used tRNA will go back out to the cytosol so it can be rebuilt and another amino acid put on it. Right? So the E site's for exit, the A site's for coming in, my P is for placement of the next amino acid, and then my E is for the exit of the used tRNA. So, and this process is called elongation. So elongation is the growing polypeptide strain. So this will continue until a stop codon is reached. Now, a stop codon does not make an amino acid, right? But what it does, it has what's called a release factor. When the stop codon is reached, it causes the ribosome units to fall apart. And at that point, translation is over. So remember, at the end of translation, I don't have a protein, right? So at the end of translation, I have a polypeptide. Remember, polypeptides need to be folded into a 3D conformation. Remember, that's called conformation. Right? So it needs to be folded. So now I have a polypeptide, and that's a primary structure. So it needs protein modification. So most proteins, as they're being made, or all proteins, will start to spontaneously fold because they have hydrophilic and hydrophobic regions, and they're in an aqueous environment. If the conformation is based on spontaneous folding, then they can be made by bacteria really easily. Right? Some need additional proteins to help fold and edit. Those are called chaperones. Right? Or if they're going to be membrane proteins, they need to go to the ER. Right? And remember, bacteria does not have an endoplasmic reticulum. So they may need lipid modification. Lipid modification would be for like a membrane protein. They may need glycosylation. So glycosylation is right, and so this needs to be made by a eukaryotic cell. Right, so or it can need phosphorylation, right? Eukaryotic cell CHOs are kind of the most abundant of those in the industry. Those are Chinese hamster ovaries, remember? Right. So all start is free ribosomes, like I said, and if it has a signal peptide, it'll go ahead and bind to the ER. So that is quick and dirty transcription and translation. And I'll talk to you soon.